these efforts with natural variation in genomics. We focus on new crop and cover crop varieties with root systems tailored to the local environment to enhance drought tolerance, efficient nutrient uptake, and ecosystem services towards sustainable agricultural goals. So with that introduction.
Thank you, John. I appreciate it. Thank you so much for the invitation. Thank you, Liz, for everything. It's a pleasure to be here today. I'm going to do my best to read the room and try to dial this in. If I'm talking in an area and that doesn't make sense to people, you actually can stop and ask me a question. But as you can see, we're going to be talking about roots today. And I will hope at minimum to entertain you with lots of spinning videos of roots. But so as John said, I'm a principal investigator at the Danforth Plant Science Center. We're just up the way on Olive and Warson, just about 10 minutes from here. We're a nonprofit plant science research center, but I do have affiliations here with WashU, including students. I'm most closely associated with the PMB program, plant and microbial biology. So I've been here actually just 10 years almost to the day, beginning of September, and the lab basically has three sort of major strands. We develop phenomics tools that allow us to study root systems, because as you can imagine, roots are pretty hard to study and therefore they are understudied. And then the idea is with better tools, we can learn something more about genetic and mechanistic basis of how roots grow, how they interact with the environment. And this third one is really enabled by the first two being successful, which is really trying to apply this to sustainable agriculture goals. So I'm going to probably focus most heavily on number one today, just like what are the possibilities of studying roots, but also we'll touch on a little bit of genetics and hopefully give you an idea of what we're really trying to do to move the needle big picture. Okay, well, so why do you choose to study roots? I mean, for one thing, we're scientists. Scientists like hard problems, and this is definitely a tough problem. But the fact of the matter is, it's hard to even study a plant and just say you're studying a plant and ignore the roots. I mean, they're really the foundation for everything we see above ground. And more and more we start thinking about our soil, the microbiome in the soil, and how it regulates major processes, nitrogen and carbon cycling, and roots really and their exudates, right, which are the carbon that they pump into the ground, drive a lot of these processes. So when we think about what plants can do or ways that we might be able to improve our crops, really roots are at the heart of it. No, roots are at the root of it, actually. So and so the lab in some form or fashion touches on all of these different areas. Okay. So we often I like to use this context or perspective, at least, to really think about the Green Revolution, right? So Green Revolution was an incredible feat of humanity by many accounts. It certainly produced a lot more food in a small amount of time and saved large amounts of people from starvation. And so we see this idea of an idiotype or an ideal wheat plant here, and you can see all of the above ground traits. And this is a classic paper, and there is a little picture of a root system here, but it's like this little tiny. We've basically been ignoring roots in our agricultural systems and certainly for breeding, right? And what's happened is our modern crop cultivars rely on what we now know. I think it's basically consensus, are really unsustainable inputs, in particular phosphorus, synthetic nitrogen, and water. We've also de-incentivized them to form alliances with microbes, right? If the soil is awash in synthetic nitrogen, the incentive to trade some carbon for some nitrogen fixed by bacteria is reduced. And we've made smaller root systems, and these root systems actually aren't that efficient. So about half of the nitrogen that we apply goes into the Mississippi River in these parts, or goes back up into the air as a greenhouse gas. So sort of modern agriculture has really exacerbated this nitrogen dilemma. So this is a phenomenal number, but in factories like this Haber-Bosch process, right, since the 50s, it's really a post-war thing. So what are we going to do with all this ammonia? Well, we can make a lot of N. So we've doubled the available N on planet Earth in about 100 years. I mean, that's a phenomenal thing to think about, right? We have really disturbed the cycles that have been going on for billions of years. And it uses a good amount of our energy supply, and just the production of it generates a lot of greenhouse gas, but also has a lot of other negative environmental consequences, right? But we need it. It also drives socioeconomic equality, right? The haves, like here in this country, we use too much, but where it's actually most needed, where there's the biggest yield gaps, it's often not affordable or just not available. Okay, so what do roots have to do with this? Well, roots are the things that are sucking up this nitrogen, right? And so that's where we've started. And so there's this concept of root system architecture, which I won't go into much detail, but basically like how roots grow, when and where they grow. Do they grow deep? Do they grow shallow? Do they make lots of fine roots or lots of big, thick roots? This really does affect their ability to capture resources, right? So in a simple example, a corn plant that puts its carbon energy, we have this term rhizoeconomics. It's like the economic part is your leaves are making carbon. What do you use that carbon for? Do you grow a root? If you grow it, do you grow it deep? Do you make a lot of branches, or do you keep growing a big, thick taproot, right? And so early on in the season, we had a drought like this year. Our field suffered from it. If you have deep roots, you avoid that drought. If you don't, you suffer if you're a plant. Later in the season, it really starts to interact with nitrogen, at least in our current agricultural systems, right? Because we apply this nitrogen just as urea typically on the top of the field or very shallowly. That N is extremely water soluble. So every time it rains, it dissolves and that water moves down through the soil column. And if it moves out of the rooting zone, it's not going to get captured. It's going to wind up in the Mississippi River, or it's going to wind up as nitrous oxide, right? But if we have deeper roots, basically these roots are in contact with that N for longer and they can absorb more of it. That's the hypothesis, at least, right? And so that's what we're trying to test. There's an additional piece to this, which is, we're thinking a lot about solutions to pull carbon out of the air. This is what plants do. I mean, that's literally their purpose. They pull it out of the air. A lot of that carbon, 40 or 50%, or in some instances more than that, goes down through the root system, right? It's used to grow roots, or it's exuded to change the chemistry of the soil, or it's traded with microbes. And so when we think about that carbon going down, that's great, but we want to keep it there. We want to sequester it. And it's pretty well accepted, although there's not enough empirical evidence, that the deeper that carbon is deposited, either through root biomass or through exudations, the more likely it is to stay down there, right? Because in the end, it's all that matters is the delta. What goes down, what comes up. We want a bigger delta. We want it to stay down there. Okay, so what's the problem? Why haven't we figured out how to make roots work for us, or at least how to make them more efficient? I mean, I think it's pretty obvious, right? The classic root work was literally archaeology, basically. And there's not a better, hey, is that you, Antonio? That's you. That's Antonio. So, I mean, there's not a good way to do roots, right? And so what we basically have is a kitchen sink approach, and I'll show you a few of the technologies and tools. All of them have caveats, but they have good uses and uses that aren't so good. It depends on your question, depends on what aspect of the biology you want to study. And so we try to kind of string them together in a way that allows us to go from discoveries in the lab to like application in the field. Okay, so we developed a system. Actually, this was a postdoc project, which this might spin, maybe not, but it's basically a three-dimensional petri dish, right? So we grow these things in big glass jars. We can grow a corn plant in there, and this should be spinning around. But they sit in these water tanks with these big cameras, and what we get is like a 3D view of this, right? So we published a lot of papers. This led to a really nice collaboration with Ken and Marshall Ledger, a student at the time, and I'll tell you a little bit about some new results on that, about how that project has grown a little bit using this main system. And so the really nice thing about it is, instead of getting this flat two-dimensional view, like if you grew roots on a petri dish or you dug them up and they get kind of floppy, we get them in like in situ. It's a fake media. It's not like soil, but it's in situ, right? And we get this, we can make three-dimensional reconstruction. So now we can measure like everything about this complex phenotype. And the better we can measure complex phenotypes, the better we can like understand how they work biologically, the better we can understand how the genetics work. So just to give you an example, these are two maize inbred lines, like ones that are actually used historically in making hybrids for production. One's Missouri 17, the other one's B73 from Iowa. And of course, the root architecture of a corn plant is basically the same early on. It makes a primary root, and you got one root. But then you can see that these two genotypes make very different decisions on where to allocate their carbon. B73 likes to make kind of that one long primary root, then it starts making a lot of little lateral roots. Missouri 17 likes to grow these what are known as nodal roots, and you can see it kind of grows these long, thick roots. And again, this has real implications for in real soil of like how well they can capture resources. And it depends on the environment, right? It's not like one's better than the other. It's all contextual, right? It all depends on the environment or the agricultural system they're growing in. But we really want to understand like temporally what's happening. So basically, yeah, we're able to use this system and a series of these 3D models, and each new color here is a new time point. So we can actually start to track. I'm maybe regretting putting this in, but hopefully you get the idea. Okay, so these are 3 different root traits. How long are the roots relative to one another? Do we have a lot of little ones or several big ones? And then like what angles are they growing at? And yeah, there's another angle trait that's like meaningful to somebody like me, right? The idea is we can start to really track how these different patterns change over the course of development, right? And so this is just a tool that wasn't available before this. And so we can really start to get a fine-grained understanding of like how they're allocating the carbon. And then if we perturb the environment, we can get at that environmental response, right? That could be biological, it could be changing nitrogen or water. So it's just a tool that we have in the toolbox, right? So one of the projects that we've been using this heavily on is Ken's plant genome project with NSF, right? This again was spurred from the initial work trying to understand the difference between some weedy rice cultivars and cultivated rice, and weedy rice can outcompete cultivated rice in the field. And one of the reasons is probably it has a bigger, better root system, and it steals all the nitrogen, right? And so Marshall did some nice work that led to us joining this grant, and now we've got like this really large scale project to look across lots of different weedy rice accessions from basically around the world. I guess there's some missing from Africa, Ken, but maybe those are hard to get. Anyways, from around the world as best we can. And then, you know, we're basically doing this time resolved phenotyping of them in the gel system to really understand, like, do all weedy rice roots, depending on, you know, like even from around the world, converge and have the same traits? Do they outcompete cultivated rice in different ways? And then are the same genes involved, right? Are the same genes being selected on? And so these are questions that we hope to be answering in this project. So this tool allows us to basically like start to reasonably answer some of these questions. So the thing perhaps we're best known for is X-ray imaging. So we have actually 2 instruments in the lab. So these are both developed for, this one really for aerospace, like industrial, like if you make airplane parts, you want to scan them occasionally and make sure you don't have a crack in it, but it's nice and big. So you can put like a whole corn plant and a pot in it. And then this one here is more developed for geology. We've co-opted it for plants. It's an X-ray microscope that allows us to look at really, really tiny things, right? And so these are here in the basement at the Danforth Center. If you ever come visit, I'm happy to take you down there and show you. There's a coffee machine. It's good. It's good stuff. It's a good tour. If you just want to like eye candy, check out the Fig Share for Keith Duncan's paper. It's just got really, really cool videos, but I'll show you a couple of them. Right? And the reason these instruments are so important is because obviously to actually see through the soil, okay, it's one thing to grow the plants in a gel, but we want to like look at them in the soil. And also a lot of plant development and processes happen internally to the plant. And so the classic way is to kill the plant and slice them up. And obviously we can't look at growth, we can't look at dynamic events in the way we'd like to by doing that. Okay, so this basically gives us this multi-scale view. So this is a sorghum root crown that we dug up and washed, in this case, and scanned. So we get this really nice 3D model, and then we can take parts of it in the X-ray microscope and like actually look at how the cells are organized in the root tip and how this is the nodal plexus. So these nodal roots like emerge at the base, and you can see one of them, that round thing on the top, is like the root coming out. So we're very interested in how roots develop and how those developmental patterns affect the overall architecture, right? Like the macro structure is just a bunch of little microprocesses happening all the time, right? So how heterogeneous or homogeneous those are is like really important to this macro property of architecture, which is like the functional thing that we care about. Right. And then so you do a lot of image analysis and sort of computer vision in the lab as well, because we have to turn these cool looking like videos into data. And to do that, we have to like train a computer as to what our eyes natively see. So this is basically, we can count every cell in this root tip, and the colored things are like different tissues in that cell, right? So it gives us like really incredible ability to sort of do developmental biology. And a similar thing here, right? This is, I don't know if you're familiar with Soteria, but it's like millet, basically, right? It's a food. And so this is a meristem, right? So you see these millet things, it's beautiful, like any grass outside. This all starts as this little tiny microscopic thing called a shoot apical meristem, or in this case, an inflorescence meristem. And again, we can see all of these pieces will become like branches later. And so we can look really early in development with this system. Okay. So hopefully I've impressed upon you some of the technical abilities we have. So like, what are we really using this for biologically? Well, getting back to this idea of root system architecture, right? So we can see these are different maize inbred lines, and they were from mostly the US. The CMLs are from Sept breeding programs in Mexico, which is the world center of maize breeding. This guy's from Texas here, this big one. WCB 73, Missouri 17. There's a few from Thailand in here. And so you can just see the variation, right? Our eyes are really good at picking this up, but how do we turn this into data, right? That we can do science on, that we can do genetics on. And so the lab, basically we dig many thousands of these every year, like probably I'd estimate 20, 25,000, right? Since the lab has been around. Okay, so like, what do we do with these? We have some ways. You can put a ruler up to them, right? And there's a 2D image analysis tool that we use, but these are 3D structures. So it's like perfect for the X-ray, right? We can scan these things in about 3 minutes. We don't have a robot to do it, but we've got like the bucket brigade, right? And we just go through these things, and we've scanned thousands of years. So we have this really nice 3D model. How do we turn that into data? Again, image processing, image analysis. We've gotten quite good at this. So we can really extract like a lot of information. In fact, we've teamed up with computer science here at Taoju, just across campus, and basically can digitally dissect a really complicated root system. We know which lateral root is connected to the parent root, and how they're organized on the root crown. And okay, I mean, why do you care about that? I'm not going to get into too many details, but this gives us a lot of leverage to understand these genetics and developmental processes that they do happen at this like very small scale, and they have implications for the larger scale that really is important for the functional traits. So I'm skipping a lot of science here, but basically what we've been able to do, there's really actually only been a handful of genes like known that control like maize, really any crop root system, right? Like how they really grow in the soil. And we were able to use these tools that I'm showing you to identify one. It's this mutation called rootless. And one of the main ways that we collect data in the field is taking soil cores, right? This is a Giddings soil core. This is at our field site in St. Charles. And Alex Liu's a student here in WashU in the PMB department. This is his project. And so we basically been able to identify a gene through genetic like approaches like GWAS and like mutant analysis that allow us to actually manipulate the depth of the root system in the soil. So getting back to this idea of like, we want to make deeper roots because we want more carbon sequestration. We want them to be in that nitrogen zone for a longer period of time. We actually now have our hand on like a tuning knob that allows us to change the quantitative trait of rooting depth, right? And so you can see that here. There's a 1 meter long soil core, and you can see that, up to about 60 centimeters, maybe 70, how you squint it, we get more roots down there. So then again, the lab does we're mostly a field lab at this point. So we test this thing out in the field. So typically business as usual around this part of the country, for 200 bushel corn to be like 150 to 200 pounds an acre of urea. added. It's a lot. Again, most of it doesn't get captured. So if you grow like a normal corn plant out there, you can see what the yield is, like in green on the left. And if you just have different root genetics, just one gene, one mutation, right? It's just change the expression a little bit, change how the roots grow. That's a really major functional effect. And additionally, like, let's say, well, let's add half the amount of nitrogen, right? Can we do this, make a more nitrogen use efficient plant, but actually use less nitrogen? We get about the same amount of yield as with the non-mutant in high end. So this is good. This isn't a definitive study. I didn't solve like the nitrogen dilemma or, this is preliminary, but it's like really promising. And this was really only enabled by some of the tools I was showing you before. Like we really stepped through many, many years to get to this point. So this is how we're using these cool tools that we have. I put this in, I don't know, maybe because it's a little more accessible to people, but this is obviously isn't a root. This is a grain head of sorghum. But this just, maybe you can appreciate, this is a hard thing to measure. We can collect these, we can go to the field, and we collected 1,000 of these, right? There's a huge variation in sorghum grain head, but how do you measure this, right? If I give you a caliper and a ruler and a pair of calipers, like it'd take you a long time even to count the seeds. And with x-rays, we can basically scan them. And this is some variation. These are different like races of sorghum that are used traditionally in breeding programs. And you can see there's a huge amount of diversity, right? You see this stuff like this thing, that's broom corn, right? That's sorghum that has like a big, kind of looks like a broom, and you cut it and flip it over and put a stick on it and sweep the floor. So, and then there's that, and then there's these really tight seed heads, right? That are bicolor things. So anyways, we can essentially use the same rinse, wash, and repeat, but we have to make new traits for seed shape and for the branching structure of panicles, but it's the same basic ideas with roots. So this isn't just, these tools aren't useful just for roots, but really any like complicated structure that's otherwise hard to measure. It's really good for the phenotyping, right? And that's kind of what we do in the lab. Then of course, in the end though, we want to be able to see these roots in the soil, right? And so we can't bring the field into here, and we can't bring the X-ray out to the field. We've tried that. We've had projects, really difficult. So it's not quite a viable like field phenotyping medicine. We can grow them in pots, little pots, big pots, and watch the roots grow over time and measure them, right? Additionally, things like, this corn plant by this stage has an ear, right? And that ear's in here. And actually I can tell you how many kernels are possible on this ear before you can even see the thing, right? If you squeeze it and you know what you're doing, you can feel it. But basically we can start predicting, and we can start to understand how, for example, drought is affecting developmental processes much earlier on. By the time the ear pops out, it's done, right? We miss the relevant like time point to understand that environmental interaction. And so as scientists, there's not, we can't make a lot of progress like that. So one of the main challenges really though with roots and soil, the X-ray scan doesn't produce like that. It produces like a 3D volume stack. So you're basically looking at it like one slice. So okay, you get a 3D volume, you take these digital slices, and these are just different media that we're growing soil or soil-like substrates that we're growing the plants in. And each little arrow points to a cross-section of a root. So it's like a real needle in the haystack kind of a thing. So again, and what we need to do is teach a computer, like, here's what's a root, here's what's soil. We have to like define that for the computer so that we can say, show us the roots or show us the soil and separate them. That's the only way we can measure them, right? It's called segmentation. So we spend quite a lot of time doing that. We teamed up with Tao again, and our machine learning expert who's now at Google. So that's why this paper's been in prep for several years. It's great to work with talented people, but all the computer scientists wind up at Google eventually. So anyways, but we made a lot of progress here. This is a soybean root system. And basically, it's just a cool animation to show you that we can effectively remove the soil, leave the soybeans, and we can, count the rhizobia on it, right? Or the nodules, right? That are these nitrogen fixing parts of the soybean. I also regret putting this in, but anyways, there's some cool computer science stuff that we did to get here. Basically, we've got okay root system, and then with this new methods, we like kept adding more and more roots. This isn't a growth thing. This is just like better computer vision programs gives us more of the actual roots in there. So this now allows us to do what I showed you with the gel system, but now with X-rays. So we can do this in soil, right? Because no one likes this gel. It's artificial. Don't roots change with the environment? It's all true. We can definitely do good science and we can tell differences in genetics, but there's a lot of limitations to growing plants in a giant jar of gel, right? And so now with X-rays, we can start to do these similar kind of experiments, much lower throughput, but we can do them in like soil-like substrates. So basically, this is a growing soybean root system, just two different time points. You can see the heterogeneity, right? Some roots grow really fast. Some roots grow just a little. We can see some roots make a lot of laterals and some don't. So even within a root system, there's this heterogeneity in these small processes. And I think this is very important to understand. One important application is for modeling, for simulations, right? Like models can be really useful. Well, I mean, there's flies, damn lies, and models, I guess. I mean, but some models are better than others, and some models can be quite useful for hypothesis generation, understanding implications of the work. But the models for growing roots are like terrible because there's no empirical data underpinning them, or very little. So this allows us then to go back and get in this loop of like simulating root growth, simulating root environment interactions, but with data that we know is realistic, and that heterogeneity is important, right? So I mean, you can see, like, maybe you want to predict how far a root's going to grow. Root length is on the left, root volume's on the right, or like how much biomass it's going to add over time. If you take the length of the root, you might say, well, the biggest roots are going to grow the fastest. And that's like generally true, but there's a lot of exceptions, right? You can see how many roots are in this root system too. I mean, there's hundreds, thousands, eventually tens or hundreds of thousands in a single root system of like a corn plant in the field. So I think it's important to understand this heterogeneity, and we can like do it with these tools. Okay, so I'm just gonna like rolling out all the hits here. This is all the cool stuff we get to play with in the lab. It's really a treat. So what I've been showing you is the root structure. Before you see a root grow, other stuff's happening. Again, it's happening at that cellular scale. There's developmental processes, their cells are dividing to elongate a root. By the time we see it grow, stuff has already happened. One of the things that happens is you got to get energy there, you got to get carbon there. And so using this method, positron emission tomography, it's a medical imaging approach. It uses a radioisotope. So normally carbon has 12 protons and neutrons in the nucleus. This has 11, so it's unstable. So it decays and it makes these radioactive particles that are detected by these coincidence detectors here. And so what that allows us to do is feed a leaf this 11 CO2 gas, usually 5 minutes, and then we blow it off and we say, where does the plant put 5 minutes of its photosynthate? Does it allocate it to other leaves? Does it move it into how much goes into the roots? And then if it moves into the roots, how much goes into the various roots, right? We expect to see that really presage that growth data that I showed you before, right? Because again, you can't grow a root if that carbon source is not already there. So that's what PET allows us to do. And we get about a 2.5 hour time period before it decays. The half-life is 20 minutes. So it's decayed many half-lives by about 3 hours, and we can't detect it. But once we get to that steady state, we pretty much can say that's the amount of carbon that was like from that 5 minutes of photosynthate. So that's allowed us to ask interesting questions about, in particular, root microbe interactions. So one obvious example is legumes. They form symbioses with nitrogen-fixing bacteria. I told you about the nitrogen dilemma. Well, microbes can, and we're breathing nitrogen right now. There's quite a lot of it, mostly N. But plants can't use that N. They need microbes to break the triple bond and move it into inorganic forms that they can take up. That's what's happening in these symbioses and these nodules. And in return, the microbes get carbon. So we can ask questions like, well, as these, what role does it have? And how does this interaction affect carbon allocation decisions? How much carbon actually goes to these nodules? How does it affect how the roots grow? So that's what we did here. We did basically 2 days of X-ray imaging, and then we did PET imaging at 11 days. So we couldn't do it on day 10, but we did on day 11. We're trying to understand, what's happening in this process. Day 10, there aren't too many nodules, and then by day 15, there's a lot. So we kind of did it at that time when this stuff these symbioses are really kicking into high gear. And so we can co-register. We can take that PET data and the X-ray data, put them together, overlay them, and we can see where the hotspots are, right? And then we can use a image analysis method called thresholding. And then we find these regions of interest where that carbon was. And again, we just measured how far the roots go. So now we get a correlation. How much carbon is at that root tip? And then 5 days later, how much did that root grow? Is there any correlation? And it turns out it's actually pretty good. In a normal plant, it's 0.64, it's significant. So in general, 5 minutes of photosynthesis can predict how long that root is going to grow 5 days later. So that tells you something about the consistency, I think, and how plants are allocating their carbon. Now, this is in one environment, and it's in corn, and there's all kinds of caveats. But we can also see that correlation gets reduced when you have the Bradyrhizobium, right? There's a new agent in there, and now the system's a little bit more dynamic, and we're losing that. And yeah, so there you go. It reduces the correlation. So we see this as root length. It's actually even better with volume, which is a better proxy of like biomass volume, because length is like just a 1D thing, or volume, we get the 3D. Okay, so we can see the effects just by the X-ray data, right? The Bradyrhizobium infected plant starts off growing really, really fast, and then it slows down as it has to give carbon to the nodules. Unaffected plant starts a little slower, but then really has to start pumping carbon down to grow its root system to capture nitrogen that it's not getting through the symbiosis, right? And so, yeah, again, we have these things overlaid. Now we're asking the question, not what's the relation of carbon to the roots, but what is the relation of carbon to the nodules? And same idea, it's an even stronger correlation, right? The amount of carbon in a nodule is like very predictive of how big that nodule will be 5 days later, how much it'll grow. So this is seemingly a very consistent relationship. And, furthermore, there's a few nodules that are kind of off the line, but it's pretty tight on there. So it looks like most nodules are pretty much, they're different sizes and they're getting different amounts, but it's scaled about the same, right? It also seems very consistent. So that was just something that, we were able to. There's not really another way to study this relationship. It was pretty new, insight. These questions we've had for a while. We're able to use these tools effectively, I think, to understand these interactions. Okay. And then it's like, well, okay, we do see different sizes in the nodules. And what is it that makes a big one? What is it that makes a small one? So here we then leverage our X-ray microscope, and we can zoom in and literally look at every piece of tissue, every cell. These are the bacterioids. So these were plant cells that got infected by these rhizobium, and now they're basically a little factory for nitrogen and carbon exchange, right? And then our ability to do segmentation is important, so we know where all the cells are, and then this thing you're seeing on the outside is like the veins, right? So this is literally carbon's going through these veins, nitrogen's coming back out. So the architecture of these veins relative to the cells and the various layers in these nodules, we know is important. The only way to study this previously was cut a nodule, slice it into little slices, and kind of reconstruct it on a microscope. And, we made decent progress like that, but we think this will be a really impactful tool there as well. These nitrogen fixing microbes are important, but these arbuscular mycorrhizal fungi are also ubiquitous in the soil. In fact, they predate, as far as we know, from fossil records basically these nodules and nitrogen fixing bacteria ripped off a lot of the molecular processes that AMF had set up several 100 million years before. That's basically what we think. And so these fungi are actually everywhere in the soil. They're a huge part of soil health. They're a huge part of holding particles of soil together, and therefore they're a major part of how water moves through soil, how nitrogen gets cycled, and how much carbon can we sink in the soil. So we're very interested in AMF fungi, and we can use basically the same tools here, right? So we infect these roots with AMF fungi, and we can track how much increased photosynthesis, how much increased carbon goes below ground, and then how much actually leaves the root and goes to the fungi. So, we can see, like, this is obviously the most extreme example, but the point here, we do get more carbon going to the root system when it's infected with these AMF fungi. And I think of these AMF basically also like an extended root system. So what they do is allow the roots to get, these roots are pretty thin, but there's pores in soil that are like 10 or 20 microns wide, and roots can't get in there, but these hyphae can. So they can actually access water and nutrients that are inaccessible by root systems. So the plant has to trade carbon for them. But basically what we've shown in these experiments is about increases, and this is just with one strain. There's lots of AMF fungi, but this is like the lab rat of AMF fungi, Rhizophagus regularis. It's everywhere in the soil, and so it's about a 12% increase in this set of experiments we did. And then what we see is about a 6% change in the biomass. So we get 12% more carbon, but only a 6% bigger root system. So by some really complicated math I did last night, I think about 5.7% of that total carbon is rhizodeposited. So either it's traded directly with the fungi, or it's exuded from the root system, but it winds up below ground in the soil and not a part of the root biomass. And it's really important to think about that rhizodeposition, because that is like the pools that the soil, that plants put in the soil, the pools of carbon have a huge influence on whether that comes back up as CO2, or it stays down and becomes mineralized or otherwise recalcitrant and sequestered, in other words. So we're thinking a lot about that these days, because, it's sort of an all hands on deck, like at least my thinking is it's all hands on deck in terms of getting greenhouse gases out of the air. So again, because I can't help it, and I told you I'd try to at least entertain you, we've also made a lot of progress with really understanding the micro scale, like in situ, like aspects of the symbiosis. And so these are arbuscules, or those are probably vesicles, but they're fungal structures within the root. Okay, so these things actually invade the root and they kind of fill up the cell, and it's at that interface that carbon and phosphorus and nitrogen are traded. And so different fungi are going to sort of have a different hyphal architecture, both in the root and outside of the root. So I was showing you what was in the root here. Here you're seeing the spores, and hopefully you can see these thin strands. These are the hyphae. So there's like 10 or 20 microns wide. And these things are literally just transporting nitrogen, water, and phosphorus and other things all around the soil, in and out of the root. And so we can really get that hyphal architecture. And that, I think, has a lot of impact on how we interpret, or at least, basically, we can have hypotheses from the PET X-ray data and sort of evaluate them with this system. Okay. Robert, yeah.
When you're setting up these experiments, is your soil sterile other than the microbial?
Yeah, the question was if the soil is sterile when we do these experiments. It depends. Sometimes we use actual reconstituted field soil that is not sterile, but there's competition. In this case, it should be basically sterile. We didn't do a test, but it's basically like out of the factory reconstituted soil. So that's a great that's a great question. In general, we'd like to understand it in the most realistic environment, but practically speaking, there's so many microbes in the soil that doing this kind of work in real soil we'd see so many different things, it would be hard to interpret it, actually. So, okay. All right, so I zoom in. I'm going to zoom out a little. I just have a couple more sections. I'm doing okay in time. And if anyone else has questions. Okay. All right. So, it's cool to see things at that micro scale, but actually, practically speaking, We actually really don't know what a full root system of most plants looks like in the real world, because even doing that excavation, that archaeology thing in the trench that I showed you, we're just looking at these 2D planes. I mean, these root systems go down in many cases of a corn plant 6 to 8 feet, and they're at least in a 6 to 8 foot radius. So there's really no way to reconstruct a 3D shape. So we borrowed this long-standing concept really from ecology of mesocosm, right, which is meant to be a step in between micro and like the real world and microcosms, right? And so it's meant to be a semi-realistic, in some aspects at least, system for evaluating science. And so we developed it for like roots. We call them 3D root mesocosms. That's you again, Antonio, isn't it? I'd know those genes anywhere. Okay, I think that we put you to work during the rotation. That's good. So anyways, this is a lot of work, but basically we build these giant boxes, and they're big enough that the root systems can basically grow freely, right? The problem with growing plants in a pot, one of many, is that they're shaped like a pot when you take the roots out, and like the root system architecture isn't that so meaningful, because they're like just kind of like a wad of roots, right? There's probably gardeners in here. You take out your pot bound plant, there's just roots at the bottom. The architecture doesn't really matter. It's just like absorptive area at this point. So we really want a way to functionally test architecture and kind of know what a real root system looks like. And you can't do that in the field, and you can't really do it in a pot. So that's where we're at. So it's not a complicated concept. I thought of it, right? So it's not that complicated. I'm not that creative. So we basically just built a big box, and we put a frame in it, and basically put the string, just fishing line. And the purpose of that is to hold the roots up, right? So they're not perfectly in place, but they hold up pretty well. This, by the way, was a switchgrass plant, if you're wondering. I forgot to mention that, but that's switchgrass. And then we fill it. We've used field soils before, or field-like soils, but they're a real pain to work with. They have living stuff in them. So we often use this calcined clay, which is turfus. So it's got some like chemical properties like soil, but it's easy to wash off roots. That's the main thing. And it's basically sterile. And then we can add microbes as needed. So we basically just grow a little corn plant in one of these. And then we also added a bunch of sensors. So we want to kind of know what's happening with the environment. We want to know in the real soil, which you don't get in a pot, there's a big temperature gradient, right? The amount of oxygen is different as you go down in the depths. The moisture is different, right? And so these are sensors that measure moisture, that measure water, measure root respiration, can measure oxygen. And we basically get like a 3D view of that along with the root. So we grow these things to maturity, and then we just open the side of the box, and with fingers and brushes and a shop vac sometimes, we suck all the turfus out, okay? And so what I'm showing you here are just same plant, switchgrass, 2 genotypes. One is adapted to lowland environments, and one is adapted to upland environments. And they grow their roots really differently. They make very different decisions on how they grow their roots, because they've adapted to where they need to be, right? And so this is something we can see in real, we can see like the whole thing here. This isn't a little seedling. This isn't a cross section of a trench. It's the whole thing. So how do we turn this into data? We borrowed this method structure from motion, or photogrammetry, which uses a lot in remote sensing, but we basically applied it for a new purpose here. So what you're looking at is like this shed that has random patterns on it. Actually, Tyler's son, a little kid, I don't know, he's 5 or 6, and he had this splatter paint all over the wall. And the reason is we want random patterns. All these green things is one camera, a view from a camera perspective. So Tyler went around with a digital camera and just took thousands of pictures. And in each picture, there's a random pattern in the background. And what modern GPUs can do these days is match up every single picture and decides which ones go together. So it can then predict the camera view that it came from. And once you can do that, you can use various 3D reconstruction methods to generate a point cloud. So now we get a 3D model, just like we got out of the gel, basically, just like we got out of X-ray, but it's like the whole root system. I mean, we're missing some roots, okay? So the finest roots are hard to get. And, you know, it's not perfectly in place, but this was pretty novel. So you just want to look, for example, at differences among, you know, some of the big agricultural plants or bioenergy plants. I mean, you can see them here, right? And then importantly, we can measure them pretty effectively, right? And then the thing that we're still working on, so I haven't really made a lot of progress here yet. This is obviously just a concept and a glowing video, but this is the sensor data, in this case, matrix water potential, which is like how hard the plant has to pull to suck up water, red being hard, blue being easy, and then temperature. So we get a temperature gradient in these boxes. And what you're looking at, the thing that showed time, oh no, there it is, yeah. So you can see these are just days. So these things grow 2-3 months. And so we basically can impute the 3D data, and we kind of know how the root system architecture is relating to these important environmental variables as they're changing. And so we've done a little more on this, but it's super preliminary, so I didn't mention it. And also it may not be that interesting to you, but anyways, it's a really nice system that's sitting in between the field and the lab work we do. Okay. This should just be 5 more minutes. Okay, this is a little moving actually more to applied work we're doing. And in fact, this is where a lot of the lab efforts these days are. So thinking about making a better root system for corn, I mean, great. We should almost definitely be growing less corn, but that's not going to happen anytime soon. I would love projects to work on perennial agriculture with Allison Miller. Many of you probably know her at the Land Institute. These are awesome ideas, but it is not going to happen anytime soon. And it's like we're in a climate emergency, in my opinion. So we have to do sort of what's the most direct thing that we can use our research platform for. So you think about the grain belt in the summer, it looks like this, horrifying or wonderful, depending on what your perspective is. I mean, it's amazing, right? It's remarkable for sure what we've done to the landscape. But this is what it looks like in the winter. For some reason, we don't put anything there. We know we have lost in some cases half of the A horizon, the good topsoil in the Midwest, and soil takes a long time to build. We can't replace this, and once it's gone, we can't grow plants. I mean, we're in deep trouble, and one of the main reasons is because there's no living roots. So like principle of soil health is living roots, and across 150 million acres of Midwest farmland every year, there's nothing. Okay, so cover crops, right? I mean, there's also secondary crops and winter crops, but this conceptually, right? Growing plants in temperate climates like ours over the winter is a good idea. And the really nice thing about cover crops is we can think about these different ecosystem services, right? And every cover crop won't do all of them, but with cover crops, we can leverage genetic and phenotypic diversity, plant species diversity, because there's dozens or hundreds of different plants that we could potentially grow as cover crops, and they all can do beneficial things, ecosystem services. So what we're trying to do now, this is just really understudied area. And what we're trying to do is like figure out what are the pinch points, like why is this the case? And there's some known reasons, right? There's some evidence and some mostly just like common wisdom, which probably isn't exactly true, that there's big yield drags with it. It does take extra work. It costs a farmer money. They're business people, just like, any other business person. So they may want to grow cover crops, but it's just not practical. So we're really trying to find like, what are the pinch points that we can address biologically? Okay, so that's what we're focused on. And the first thing we have to do is just survey the roots and start getting these things, growing them out in the field, getting to know them. So this is a really cool picture, but basically all these things were grown in like pots or pipes, right? And it's just an N of 1 basically. So this illustrates the diversity, but there's not a lot of empirical data on the root system architecture or functions for these cover crop species, both the ones that are currently used in some cases and ones that are just potential, like have a lot of potential. So at our field site, we started to do like a lot of just the basic work, like to build that foundational knowledge base. So what you're looking at here are several cover crops which are used. The major one is cereal rye, but we're also have several other grasses, some clovers, and some brassicas. Those are kind of three of the main categories. There's broadleaf, I guess, would be the fourth category. But we can just see a lot of variation in how much shoot growth, how much root growth, and then the relative relationships. And there's a whole bunch of other things that we're measuring here. So this is what the field looks like. This is in St. Charles. And if you ever want to come see it, I'd be happy to show it off. We're about to plant. The thing about cover crops is like, and field work is that basically now we have like 12 months of the year field work instead of 6 months. And that kind of sucks, but we just are, getting yield and getting our corn out. And then we immediately are planting cover crops. So these will grow until it gets cold, mid-November or December if we're lucky. They'll basically stay and just like kind of like freeze in place. And then in March or whenever it starts to get warm, then they start to really rapidly grow. So this is what they look like this spring. Some things did well, some things didn't. It's basically a 13-acre plot. So we have like 60 by 60 foot replicated design. And in this case, there are tons of stuff. We did like as many species as we could fit in there. And then of course, in the end, right, taking first principles from ecosystems, right? You don't see ecosystems that are monocultures, even cover crops, right? They're all mixes. And so we've been trying to like kind of rationally design different mixes that have different ecosystem services that might be compatible, that don't have incompatible root systems, that aren't competing with one another. And we're trying to evaluate those out in the field. And so, one of the things we're also focused on is what does it do to the corn? So in this case, we grew the exact same Bex hybrid, I don't remember the name of it, XYZ123, exact same uniform hybrid monoculture. And you can hopefully see, right, this yellow stuff with the tassels. You can see differences in plant height. You can see differences in how green they are. This is after 1 year of growing cover crops, and we planted the same stuff in there. So it does have an effect on corn. It has an effect on how fast the roots grow. It does, there's some nitrogen interactions. This strip here is conventional. So this is all planted no-till. And then we got this strip and that strip, which is tilled and just planted conventionally with fertilizer. So anyways, this is a big experiment. And the idea of it is that we're basically trying to study, changes to the soil, changes to the interaction with corn, and then changes the cover crop traits themselves, right? It doesn't all happen at once. Over time, each of these different species will basically start to take a different trajectory with the soil, how it's changing, reducing bulk density, increasing the porosity, gas exchange, that will change microbial diversity, and so on and so forth. So this is year one, and I'm gonna keep this going as long as I can keep it funded, basically. So we have, 5-10 years from now, hopefully I'll be able to tell you about what we've learned like over that period of time. So we're measuring, microbes. Didn't talk with you about some of our microbiome work today, but we are doing some of that, soil properties and root traits. So, okay, with that, if there's time and interest for questions, that'd be great. I'd like to, of course, thank the lab. Again, if any of you ever want to come visit the Danforth Center, it's a really awesome place, and you can look me up and I'm happy to host you, show you around. There's a lot of great science happening there. And it's a really good community. It's a pleasure to come to work every day and do exactly what I was telling you about today. That's we all have a love of science, and so any interest in it, I'm always welcome to oblige you. So thanks again for your attention. I appreciate it.
Okay, that was a great talk. I loved all those figures, rotating figures. Very impressive. Do we have any questions for the speaker?
Hey, thanks so much for that wonderful talk.
Thanks.
I kind of have more of a technical question, I guess. It seems like the amount of data you guys must be generating from this genomics work must be insane.
Yeah.
Just give us an idea of sort of storing this data, how much do you take up?
That's a great question. We've collected nearly a petabyte of data from mainly from the X-ray systems. A lot of it's big in one sense, but it can be compressed into a CSV file. But the image work, those 3D volumes, about a petabyte. we've got a fantastic data science center, but and they basically run, we've got several like storage, like RAID storage units. If I'm being honest, it does keep me up at night sometimes, right? I mean, we basically can't back all this up on the cloud. It's too much. Some of it's in Amazon Glacier, which is a lot cheaper, but that's stuff that we don't plan to actively use. This is a big problem in the field of phenomics. Maybe you're aware of it. It's, we're not the only group that has this problem. Not only is it is storing stuff we're actively using a problem, but when we publish a paper, like we need to make the data available. The code's easy, we put it on GitHub, no problem. But putting the data somewhere, there's no place to put it, and no place we can afford to put it actually. So I don't have an answer. We're just kind of like muddling by and hoping no disaster doesn't strike. So yeah.
Thanks, Chris. You spent a lot of time removing the soil, for example, so you could see the roots. And I wonder, so for example, some of the figures that you show where you're quantifying like, oh, these roots grew faster, they grew slower. Yeah. Do you take into account somehow the soil around the root, that specific root, for example, and how do you model that or take that into account? That's
yeah.
Thanks.
Yeah. So I left all this out, unfortunately, but we're doing a lot of work with soils. The really nice thing about these x-rays is actually we can scan, they work just as well. So you're right, we can remove the root and look at the soil, right? So one of the things we're doing is, so I showed you what we're starting with the cover crops, which is like from 0 to hopefully 5 and 10 years. But we've worked with Chuck Rice, who's a soil scientist, as a grad student, and he runs this experiment at Konza Prairie, which is like one of the, I think it's certainly the largest tallgrass prairie remnant left. I'm pretty certain of that in Kansas. And he's had this long-term experiment, 30 plus years now, it's like 34 years, where they've done no-till conventional urea versus and manure. And they've just like, how has the soil changed? How has corn yield changed over time? And so we've basically been x-ray scanning the soil and another level of soil, which are aggregates. So all these soil actually made up of really small things called aggregates. And so long story short, we see huge differences in porosity, soil organic matter, just like the structure of the soil. Have not yet put the chocolate and the peanut butter together just because it's like really complicated. But that's exactly what, in 5 years from now, I hope I can say, yeah, we're doing that. So it's a great question. Thanks. And then he's and I'll be at the reception too. So if there's not time, sorry.
This is really interesting to me, particularly the differences among different cultivars in terms of the root structure and volume and all the other sort of characteristics. And a number of those cultivars were grown in many different parts of the world. And I'm wondering if you looked at it, was it possible to pull out some principles about the various kinds of root structure based not only on soil type, which kind of follow up to the previous question, but also in terms of environmental variation, other sorts of things.
I hope that is possible. I hope that's possible. My people have made efforts at like various classifications for root systems. And I think like while they're interesting, I have not felt that there's enough real empirical data to, they rely heavily on models and simulations, which I have concerns about.
Now you follow up, how much phenotypic plasticity is there on root structure?
Phenotypic plasticity is a genetic trait, in my opinion. So there is genetic variation for the amount of phenotypic plasticity. So I didn't show there's a lot of slides I didn't show, but so a basic thing like response to nitrogen. And I showed you those different varieties of corn. Some of those, if you give it a low amount of nitrogen in the field, like half the amount, grow very similarly. Like they don't change that much. They're pretty conservative. some will change dramatically, like twofold increase in the root biomass, right? So, and you can't answer that in general. Every root system is gonna have some environmental effect. There's always an E effect, right? But it's that G by E effect, actually, which is something that we can actually map genetically, right, using phenotypic analysis and like GWAS, for example. That's a really interesting area. And so we do some of that, but I can't give you any like, like pearls of wisdom in this case.
Okay, so we're about out of time. You can join Chris and everyone for receptions. We should be downstairs outside, someone across the hall. And let's give Chris a round of applause.
Thanks, y'all.
Yeah.